Hello and welcome to this edition of Work of a Scientist. Today I will introduce you to Francoise Barre-Sinoussi, the co-discoverer of HIV. Francoise spent her career working at the Pasteur Institute in France. She was born in July of 1947 in Paris, and from a very early age it was obvious that she had the mind of a scientist. It was her best subject in school. As a child, she loved spending time outdoors. She was always making observations of the natural world. She spent countless hours wondering about the behavior of animals. She was even fascinated by insects. Although she wanted to attend medical school to become a doctor, she decided instead to pursue a career as a research scientist. She got her undergraduate degree in natural sciences from the University of Paris. While still a student there, she looked for a research lab that would let her gain experience. After many months of looking, she finally found a position as a volunteer researcher with the Pasteur Institute. The particular lab she worked in focused on the relationship between retroviruses and cancers in mice. Very soon after starting here, she began working on her PhD project in this lab. She studied a molecule that could inhibit reverse transcriptase, an enzyme that turns viral RNA into host cell DNA. She was able to show that inhibiting this enzyme could slow the progression of disease in mice, and she received her PhD. Shortly after this, clinicians around the world were noticing a new agent was causing an immunodeficiency virus. In early 1983, her lab received a tissue biopsy from lymph node of a patient with symptoms of AIDS. They isolated lymphocytes and cultured them. She detected some reverse transcriptase activity in this cell culture, but it was very difficult to work with because the lymphocytes were dying too quickly. She had to perfect a protocol to keep the culture alive by adding fresh lymphocytes on a schedule. And eventually she was able to isolate a virus called LAV, for lymphadenopathy-associated virus. This was published in May 1983 in the Journal of Science. In the ensuing months, she and other researchers continued to characterize the new virus. However, there was some controversy about this. Another virus, known as HTLV, was thought to possibly be the cause of AIDS. Robert Gallo, a researcher in the U.S., was an expert on HTLV, which was the only known retrovirus in humans at the time. Human T lymphotropic virus causes cancers, such as leukemia, in humans. However, if you look at HTLV prevalence maps, you'll see that it's prevalent in Japan and the Middle East. However, AIDS was not occurring in any great numbers in these countries, so it was likely that LAV was the agent that was causing AIDS. Part of the confusion stemmed from patients being co-infected with HTLV and HIV. In 1992, Francoise was promoted to head of the Biology of Retroviruses unit. And by this time, LAV had been renamed HIV and was known as the definitive cause of AIDS. This is Francoise' sketch of science, an HIV virion attaching to a T cell receptor. This receptor is on the CD4 molecule found on helper T cells as well as some other white blood cells. Reverse transcriptase is very important for turning the HIV RNA into cell DNA. So therefore, it is a very good target for antiretroviral therapy. Francoise has worked very hard to improve treatment access. You can see in 2007, many more countries were having patients treated with antiretroviral therapy. In 2008, Francoise received the Nobel Prize in Physiology or Medicine, and from 2012 to 2014, she was the president of the International AIDS Society. She finally retired from research in 2017. NGSS connections to this relate to growth and development of organisms. And these are the main resources that were used for this presentation.